thank you for the organizers to have CARISMA present, and congratulations on your 10-year anniversary. I'm going to spend the next 15 minutes telling you a little bit about CARISMA. We are a preclinical private company that is developing engineered macrophages. And so we're leveraging the 30 years experience on CAR T and taking lessons that were learned in synthetic biology, vector biology, adoptive cell therapy just in general, the manufacturing, et cetera, and trying to apply it to a new cell type. So why would we consider doing that? And I think it's related to the fact that while there's very, very good success in heme malignancy, there's been challenges in solid tumors, and a number of different companies are addressing it. We just chose to take a different path. We're looking at the three, what we feel are three primary obstacles: trafficking to the site of the tumor, the access of cells into that tumor microenvironment. Secondly, an immunosuppressive tumor microenvironment, overcoming the challenges there. And lastly, antigen heterogeneity. We believe that macrophages are well positioned to address each of those, and I'll be talking through those as we go forward. So why macrophages? Macrophages are well suited for solid tumors. They're very abundant in the tumor microenvironment. The graph on the left shows a number of studies. These are thousands of patients. Each of those dots represents a single trial with hundreds of patients in them. And across the board, regardless of tumor type, you see that macrophages are heavily infiltrated, much more so than T cells. Secondly, the effector function of macrophages lends itself as well. The primary mechanism of killing is phagocytosis. Macrophages also can recruit other effector cells, T cells, NK cells. They secrete pro-inflammatory cytokines to warm up the tumor microenvironment. And lastly, they're a professional antigen presenter. So they can process and present antigens or neoepitopes within a cancer cell out to those immune cells that they've recruited. If you think about what the platform looks like graphically, from left to right, we have a macrophage that recognizes an antigen, a surface antigen on a tumor cell, and they identify it, grab a hold of it, consume it, and kill that tumor cell. Then they process and present the antigens back out, there's a pointer here, back out to circulating T cells, and then activate an adaptive immune response. So in essence, what we're doing, unlike the T cell approaches, which are mimicking adaptive immunity very powerfully, what we're trying to do is mimic the innate immune system and drive true adaptive immunity. So that is our platform as a whole. Now, the first step, and I think the breakthrough that occurred by our co-founder, Mike Lachinsky, as he was doing this work at Penn, was identifying a way to transduce macrophages. AAV doesn't work, Lenti doesn't work, and what he found is an adenovirus chimera called Ad5F35 that effectively and reproducibly transduced macrophages at the 60 to 80 percentile. And so that was the breakthrough of the company. I think really the formation of the company was a domino after that. The next question was, will ex vivo activated macrophages or macrophages in general traffic to the site of the tumor? And so here are three different examples. One, a tumor organoid model that had been co-cultured with macrophages, and you can see very rich antigen or macrophage density throughout the tumor organoid on the left. We have tumor xenograft models where we tail vein administered macrophages, and you can see a good overlap between the tumor mass between the tumor mass here and the macrophages and where they go. Lastly, back in the 90s, Reinhardt and Dreesen ex vivo activated macrophages and administered them. And you can see here, this is indium 111 labeled macrophages. You can see good overlap with some Mets in the tumor. So we know that they're getting there. Will CAR macrophages get there is the next question. Here's a model that we have, it's a gastric carcinoma xenograft model, where on the left are untransduced macrophages versus CAR macrophages targeting HER2. And you can see the blue bar is trafficking to the tumors, and you can see with the CAR macrophages, approximately 50% of the macrophages make it to the tumor. The next most common place to reside is in the liver, which may be useful for liver mets. But so we can see that CAR macrophages are trafficking in animal models. The next question is, are they targeting and phagocytosing the antigen target of choice? Two different ways of looking at it. The first, on the left, is control macrophages, untransduced, versus CAR macrophages targeting an antigen. You can see much higher level of phagocytosis of our target cells with the CAR macrophages. The next way to look at it is target positive versus target negative tumors. And so on the right-hand graph, you can see target positive. These are in blue. You can see highly transduced, highly phagocytosis events versus the target negative. And so we know that they're targeting. We know that they're killing. How does that occur? Here's a graph. The last time I was here, I showed a movie. I'm not going to show a movie today, but basically, it's about a 7-hour process from contact with the cancer cell, consumption, degradation, breakdown, and then a recovery of the macrophage. We do know that CAR macrophages can consume serially. And so we've seen them after the repair, they're reactivated and going forward. We also know that CAR macrophages can consume in parallel, and we have examples of up to 18 different tumor cells within a macrophage. So that we know that they're going forward and killing. We used, in some animal models, we've taken a look at what happens with tumor regression and survival. This is purely based on the phagocytosis activity, has nothing to do with the immune system, and we see the CAR macrophages extend survival up to 100 days, or 50% survival at 100 days in this model. Next, and I think a key question, if you think about macrophages in the tumors, most of them exist as M2s, or tumor-associated macrophages. Very conducive to the tumor microenvironment, make it a happy home for cancer cells and other immune effector cells. We need to go in and have activity as an M1 phenotype. We're fortunate that through our process, we're able to make M1s using our adenovirus. It converts them to M1 and locks them into an M1 phenotype. We can confirm that through heat map modeling of different genes that are upregulated, and we see that across the board, you see antigen processing and presentation, we see MHC class 1 and 2 upregulated, co-stimulatory ligands, et cetera. So we are acting as an M1 in that tumor microenvironment. That also lends itself to our second mechanism of action, which is to recruit other immune effector cells and have activity. We did ask the question, I will admit it's a somewhat contrived model, will we process and present neoepitopes? And here we have a HER2-targeted CAR macrophage. We've inserted a gene so it expresses NY-ESO-1, and we were trying to demonstrate, will it present other antigens? And in fact, we did see that with our preloaded CAR macrophage. We saw NY-ESO-1 presentation and antigen-specific CD8 cells upregulated. CAR macrophages also secrete a myriad of immune stimulatory factors, and I'm not going to go into details on these, but they're a very robust expression of different factors, which all lend themselves to both recruitment of immune cells as well as activation of bystander immune cells. So, for example, we did do a study where we looked at recruitment of T cells, and this is a chemotaxis model that we did, and we see that CAR macrophages are definitely recruiting human T cells in this chemotaxis model. So we think that should translate into the human testing. Secondarily, we've seen CAR macrophages activate dendritic cells. So bystander dendritic cells are activated, which and we've done this in a conditioned media, CAR macrophage conditioned media, and we saw activation. And lastly, we looked to see what would happen if a CAR macrophage goes into a TAM, or tumor-associated macrophage-rich environment, and we see that, again, co-culturing with some supernatant from CAR macrophages, we see the conversion of M2s into M1. Of course, we don't know that's going to happen yet in the humans, but it's directionally is where we're going. So we see antigen presentation, we see activation of immune effector cells, and we see some conversion for M2 macrophages. So where do we go from here? Last year, when I presented, we were just coming off of Series A. We raised $59 million last year. We had 6 employees, I think, and we were actively trying to take this technology and get it into the clinic. As of today, we have grown. We're 23 people, not a huge company, but still growing. We have taken our lead program, which is a HER2-targeted CAR macrophage, and are close to filing an IND. We anticipate filing the IND by the end of this year. We have also identified and constructed 2 other CAR macrophages targeting mesothelin and PSMA, and we've made those, and we're currently doing some preclinical evaluation of those as an activity. In addition to that, we're spending a lot of time on our platform looking at novel CAR constructs that are more macrophage specific. We're working on novel gene delivery mechanisms, non-viral gene delivery, and we're looking at optimizing the cell and activation state. And we're continuing to look at expansion of our pipeline. Specifically looking at our lead program, CT-0508, it is so we've made it. We are moving quite rapidly. I've shown you some of this data in a prior slide, but it's the first generation CAR macrophage. We have partnered with a number of CDMOs. Oxford Genetics has made our plasmid. WuXi is making our viral vector. We've just completed GMP manufacture of our viral vector, and we're partnered with Miltenyi on a cell product. It's a relatively short process, 7-day process. We apheresis, harvest cells, harvest monocytes. We differentiate into macrophage, introduce the viral vector, and then cryopreserve. It's 7 days from when it comes into our suite until it's cryopreserved. We are, as I said, close to initiating our phase 1 study, IND by the end of this year, phase 1 study in HER2 positive patients. It'll be a basket design where we're looking at a number of different tumor types. We'll be administering our product. First 3 patients will be on a monthly basis, and then we'll increase the size of those of the enrollment per month. We're targeting 18 patients and anticipate having enrollment done by sometime in Q3 next year. The things that we'll be measuring, and obviously how we define a successful phase 1 study, one, that we can reproducibly manufacture this, and we're very confident we can do that. Second, that it's a safe product. Third, that the CAR macrophages traffic to the site of the tumor, and we're going to measure that through zirconium-89 labeled cells and PET imaging. And then we would say that that is successful in overcoming the first hurdle for cell therapy, which is getting into the tumor. In addition to that, we'll be, of course, measuring target engagement, phagocytosis, tumor microenvironment modulation, recruitment of immune effector cells, and an adaptive immune response. We'll be looking at persistence of our macrophages. It's not an expandable population. It won't replicate and grow in the body. So we have a finite period of time. We want to know what that is. And lastly, we want to demonstrate that we can maintain our phenotype throughout the life of that macrophage. So next steps for us are IND filing, phase 1 initiation, in vitro and in vivo proof of concept on our next 2 CAR macrophages, mesothelin and PSMA. And we want to look at other disease applications, non-oncology disease applications. We're doing some preliminary work there. We want to continue to work on our platform and develop novel next-generation macrophage constructs, both CAR and CAR-like. And we want to look at non-viral gene delivery as an alternative to our adenovirus construct. And lastly, we want to explore allogeneic approaches for CAR macrophages and also explore looking at monocytes, not just macrophages.